Hao. I'm the second year's FPCTS student. So today I'm very excited to introduce my final thesis topic on validation of cough quality of life questionnaires in patients with chronic cough. My advisors are Dr. Harburg and Dr. Sugar. So first, let me briefly introduce about our study's background. The chronic cough was defined as persistent cough lasting at least 8 weeks in adults, and it will significantly impact patients' quality of life in the physical, psychological, and social functioning aspects. According to a 2022 study, the average total cost on cough-related healthcare is per patient was almost $20,095 during the 12 months before and after the first visit of the patient. And during the cost, 63% of the cost was for the diagnostic investigation accounting, and nearly 25% of them are for cough clinic consultations. Given the substantial burden of the chronic cough, both on the patient quality of life and the healthcare system, we need to accurately measure the impacts of the chronic cough. So the objective measures like cough frequency will directly measure the cough characteristics of patients, and it was currently used in clinical trials, but it has the drawbacks of it may fail to capture the broader impacts on patients' quality of life. So we may not get the patients whether their physical status or psychosocial status among the directly measures of cough frequency. So there have been more and more interest in using the PROMS, therefore patient-reported outcome measures, comprehensively evaluate the patient's health status. And both the FDA and EMA have emphasized the importance of using the PROMS in clinical trials of the chronic cough. So in our study, we are mainly focused on two PROMS. First is the LCQ, stand for the Lancaster Cough Questionnaire, and actually it was widely used as a primary outcome measure in currently. And our primary interest questionnaire was CQLQ, stand for Cough Quality of Life Questionnaire. And the CQLQ was based upon a small single center study with the IPF population, but haven't been validated in a larger multi-center cohort, and that should be like what we should do in our study. And it's worth mentioning that both of these two questionnaires have been recommended by the European Respiratory Society 2020 guidelines. Here's some of the information about the LCQ and the CQLQ. We can see that they are made up of, LCQ is made up of 90 items, and the CQLQ is 28. So they have different domains. So we can see that at least exactly the four domains here. We see that CQLQ and the social and psychosocial or any others domains which may not show in LCQ. So it may have a more comprehensive aspect in the evaluation as QLQ. It's worth mentioning that the direction of these two questionnaires actually they are opposites, while the higher score measured by LCQ means that the patient have a better health, but the higher score in CQLQ means that patient may have a worse health status. So the objectives of my study first, we will evaluate the psychometric properties of CQLQ in our cocoa cohort. And actually, it's a chronic refractory cough cohort comparing to our gold standard LCQ. And the second objective is we'll trying to evaluate the ability to of the CQLQ to distinguish patients, to distinguish the severe cough patients from the all the cough patients. And the last objective is to calculate the minimally clinical difference of CQLQ using our baseline data. And we'll use the distributional method to calculate it. So it should be a cross-sectional MID. The full name of our cohort is the chronic refractory cough cohort, and the cohort was conducted by 15 centers, 15 American Lung Association Airway Clinical Research Centers. And the patients were enrolled between July of 2017 and August of 2019. Here, we'll define the chronic refractory cough as the participant in our cohort, they have chronic cough and they take the most common treatment for cough like the treatment for asthma or GERD or upper airway cough, and unfortunately there was no effect of them, so they were defined as the refractory, and we will enroll these patients in our cohort. Here shows the data collection table. Just as the table shows that we have 2 visits apart from 4 weeks. So in the baseline visit, we will collect the consent or eligibility and the baseline medical history. Both of the CQLQ and LCQ data were collected in these 2 visits. And the other questionnaires were also collected, but we didn't include them in our study. Also, blood draw for storage or is collected, and that actually for published study, another study published. Yeah. So then I will introduce the statistical evaluation metric I use to evaluate the characteristic of the questionnaire. First, the Chromebox alpha coefficients will measure the internal consistency within the CQLQ. We'll use the baseline data, and the Chromebox alpha coefficient between 27 and 29 were considered a good internal consistency. And the evaluation will have a focus both on the 28 items of the total score and in different domains. Then the correlation evaluation will compare the LCQ total score, the baseline total score versus the CQLQ baseline total score. Spearman coefficients will be used to see whether our interest questionnaire CQLQ have measured the same characteristics compared to the LCQ. And then in the repeatability analysis, we will use both of the visits, both of data collected in both of the visits, and ICC with one-way random effect model will be calculated, and the BA plot will display the trend of in our two visits. Then the ROC curve will be generated, and we'll use the LCQ cutoffs shown here to divide our cohort into three groups with severe, moderate, and mild patients. And the cutoff between severe and moderate is 12.82. And we'll use this cutoff to distinguish to evaluate whether our CQLQ data, whether our CQLQ can distinguish these patients. And then sensitivity and specificity plot will be generated to find exact CQLQ threshold to distinguish these two cohorts. Both the intersection points and the point with maximized unit index will be found to see where to find the CQLQ threshold. Here, these two methods are both used to find the optimal cutoff to balance both the sensitivity and the specificity. And at last, we will calculate the MID using the distributional method. And actually, it's just 0.5 standard deviation of the baseline CQLQ total score. Then comes to the result part. We have 190 patients enrolled at baseline, and we'll include their data for the baseline analysis. And we have 3 patient drop off before the visit too, and we'll exclude them from the repeatability analysis. So we only have 160 patients in our repeatability analysis. Here's the table 1 summarize the demographic characteristics of our patient. So since we will compare, since we divide the patient into 2 groups, so I use 3 columns here and comparing the characteristics using the statistical test. And we can see that by looking through the p-value, the demographic characteristics in our 2 groups are comparable, and there didn't show any differences. And these are the cough characteristics we collect from our baseline. And we can see that obviously the baseline score are statistical difference since we get a very low p-value. And the cough upon waking are also different between 2 groups. Yeah. And also the phenomenon in the chest is have the p-value of 0.05. So we can see that the characteristics are apparently different between our 2 groups, but since they are both the cough patients, since we recruit patients with cough, so some of the characteristics they are. For example, the coughing at night, they're not statistically significant, but they have the cough symptom. Here shows the internal consistency results from our analysis. We can see that except the emotional well-being, extreme physical competence, personal safety fears with 4 items, and the other scales, the other domains shows a better or shows good consistency. And it's interesting that they didn't have a better have a good conduct of coefficients, and we'll remain it to our discussion part to discussion about the problem here. And here we get a correlation coefficient of -0.82 as our results. So it means that there's a strong and negative correlation between our 2 questionnaires, which consistent to our to their direction, their opposite direction. And the scatter plot I put here also shows very clear correlations between the 2 questionnaires. Here's the repeatability analysis. So we results 0.95 and 0.69 ICC value, which means that the consistency was at the moderate level. And since we don't have much high ICC, I also did a Wilcoxon sign red test to test whether we have a change between our week 4 and the baseline. So we got a -5.83 score change between 2 visits, and it has a very low p-value. So yeah, and also in the BA plots, I fit a regression line, and the slope, the test of the slope didn't show the significance. So it means that the difference between the 2 visits were not related to the magnitude of the scores, which means that the participant with higher scores, they will not show a higher or lower trend in the difference. The ROC curves shows a good result. We have 0.92 as our area under curve, which means the CQLQ was a good tool to distinguish the patients between the to distinguish severe cough patients. In our sensitivity and specificity plot, we can see that we have an intersection point at around 59, and the unit index was around 61.5. So they both have a good sensitivity and specificity at around the point A. So we'll conclude that our CQLQ is a good tool to distinguish the patient, and we can choose whether we can choose these 2 points as the threshold to things like maybe we can say that patients have a score lower than 59 will be a mild patient, or mild or moderate patient, and the patient have over 59 score will have severe symptom. Yeah. The result of the MID was calculated by the standard deviation of the baseline total score. So since we use the distributional method, the MID calculated is 7.78 here, which means that if a patient experienced 7.78 or more than 7.78 discrepancy in their scores, the change of the cough can be seen as clinical significantly or clinical importantly. So in my discussion part, so just as we see in the internal consistency table, we can see that in some of the CQ domains we have lower crop box alpha, lower than the standard of 0.7 to 9. So it's maybe because of the smaller numbers of items in these domains, since they have 4 or 3 domains, 4 or 3 items. And it may also mean that they have a heterogeneous in their items in these domains. And the moderate retestability of 0.89 comparing to a previous study shows that in their study, their ICC value is 0.89 and actually get a lower ICC. It's maybe because the phenomenon of regression to mean, and may also the research may also be that we have a longer interval than their study. We have 4 weeks, and they actually have 2 weeks, only have 2 weeks. And the high sensitivity and the specificity at the CQ cutoff of 59 shows that it should be a reliable tool to distinguish the patient from the severe cough patient. We had some limitations in our study. Since we recruit the chronic refractory cough patients, so we only have 2 patients with mild cough under our criterion of determined by one of the previous published paper. So it will limit our generality of the conclusion. So we also can't we can't evaluate the ability to distinguish the mild and the moderate patient. Yeah, since we are lack of the patient or healthy people. And the MID determining in our study was cross-sectional. Since actually we didn't have any intervention in our study, so in the future study, maybe within interventional based cohorts, anchor-based MID could be better used and support our results of 7.78 as the MID. Overall, I will still conclude as our finding will support the CQ as a reliable and clinical valid tool to use in more multi-center trials or any studies assessing the cohort-related quality of life patients. So it has potentially utility in both clinical and research setting for evaluating the patient's outcomes, and it also have a better, it also have good ability to distinguish people. It can help us identify severe cough patients who may need the more urgent treatment. Yeah, and thank you for your listening, and welcome to any comments or suggestion. Yeah.
Great job, thank you. Any questions?
I had one. I was thinking, as we've looked at these data sort of fresh off the presses, we've been working, and mostly Hal has been working very hard. But that, although it's within the same study, but I think the fact that the difference that we saw between 0 and 4 weeks was less than the MID, is it kind of fits the story in that we didn't have any intervention in that time. And so even though the difference, if you did like a paired t-test, looks like a significant difference, it wasn't greater than the MID, which, would certainly color or any kind of interpretation. So that has just struck me in the last day. So I hadn't had a chance to discuss it with Hal yet.
Yeah. Actually, that's a good question. And so we have the MID we calculated 7.08, but so for the decreasing of our scores.
Yeah, I think you had it just a second ago with the difference, and it had a Wilcoxon test.
Yeah. I made the Wilcoxon Sun-Reg test, and actually the mean change is negative 5.83, and it's less than the MID, right?
Yeah, so it looks very significant, but it's less than the MID. So it's just, I don't know, maybe another point to draw out that, you need to really have whether some metrics to evaluate what change means.
Yeah.
And not just the data itself or p-values.
Gotcha. Yes. Yeah. I actually we have like also collect the GAOC data or GACC global assessment of change. Maybe in the future or in the future works we can. I don't know whether we can use it as our anchors to determine the MID. Maybe we can talk more about that.
Sure. And I think what we actually should go back and look at, and we had it way before your time in a very rough way, is the cough diary.
Yeah. Yeah. Any other problems or?
No last questions? All right, thank you.
Thank you.
Up next, we have Jan presenting. Okay.
My boss is here. That's usually. Yeah.
Hello everyone, my name is Yan. I'm a second year master student in epidemiology and also in the CTS track. It's a great honor to present my thesis project today. I would like to first express my sincere gratitude to my thesis advisors, Dr. Rose and Dr. Jacks, for their invaluable guidance and support throughout the project. I'm also grateful to the Achieve coordinator for providing access to the data used in this study. I really appreciate it. Well, the topic of my thesis is distinguishing retest effects in cognitive aging findings from the Achieve study. First, why we are interested in retest effects. Though retest effects, also known as the practice effects, refer to the improvement in test performance while repeating technological tactics. It can be explained by the content of neuropsychological tests themselves and the context or conditions under which they are given. Factors like examining familiarity with the test, reduced anxiety, absolute memory, etc., could play roles in retest effects. Given that the magnitude of retest effect may be comparable to up to 10 years of age-related decline in the opposite direction, it can obscure risks of cognitive decline in cognitive aging studies and present a significant methodological challenge in longitudinal studies of cognitive aging. Failing to properly account for this could lead to incorrect estimates of the trajectory of cognitive aging. Therefore, specification of the retest effect is crucial for accurate estimates of cognitive trajectory in older adults. And emergency evidence has identified hearing loss as a potentially modifiable risk factor for cognitive decline and dementia in older adults, with hearing interventions showing promise in reducing cognitive decline. The ACHIEVE study, which is a multi-center randomized controlled trial, investigated the effect of hearing intervention, such as providing hearing aids and audiological counseling in reducing cognitive decline in older adults. It stated a potential benefit of hearing intervention for high-risk populations, such as those from the Eric study, because this longitudinal trial included a follow-up period of 3 years. Here, continue chat.
Suggest.
Oh. Because the longitudinal trial included a follow-up period of 3 years from baseline, during which cognitive tests were conducted at 4 time points: baseline, year 1, year 2, and year 3. We are interested in whether there was significant retest effects within the ACHIEVE. So for my thesis, we use the data from ACHIEVE study to distinguish the retest effect. Our study is aimed to investigate the magnitude of retest effect in ACHIEVE study, and specifically, we are also interested in whether the retest effect differ or could be modified by baseline hearing levels, hearing intervention, and population sources. Well, for baseline hearing levels, we compared participants with total average between 40 to 17 to participants with total average between 30 to 40. The total average between 30 to 40 corresponds to mild hearing loss, typically causing difficulty in understanding soft speech. However, PTA between 40 to 70 indicates moderate to moderately severe hearing loss, which can significantly impact daily communication and often necessitate the use of hearing aids. For hearing intervention, we compared hearing aids group to health education group, and for population sources, we compared the de novo subcohort to ARIC subcohort. Next, I'll introduce the study design. First, the data used for the analysis is from ACHIEVE study. There is no additional exclusion or inclusion criteria. The primary outcome of interest was the time-varying global cognition factor score derived from a comprehensive neurocognitive battery administered in person, but excluding the digital span backwards and logical memory tests because these two tests are auditory dependent. The secondary outcomes include domain-specific factor scores for language, executive function, and memory. The factor scores are derived from a comprehensive neurocognitive battery by using a latent variable method. These neurological battery include the following tests: delayed word recall, digit symbol substitution, incidental learning, making parts A and B, logical memory, digit span backwards, Boston naming, word fluency, and word naming. The factor score for language domain is derived from Boston naming, word fluency, and animal naming test. The executive function domain factor score is derived from making parts A and B and digital symbol substitution. The memory domain factor score is derived from the delayed word recall, logic memory, and incidental learning. The primary exposure of interest was retest fact modeled as a binary indicator, 0 for the first assessment and 1 for the subsequent testing occasions, to capture the boost in cognitive outcome between baseline and subsequent visit. To investigate the heterogeneity in retest effect, we included 3 key variables of interest based on the design of the achieved study. Baseline hearing levels, intervention groups, and population sources. The interaction terms between each of these variables and the retest effect were included in the model to evaluate whether the retest effect differed by the 3 features of interest. The demographic covariates we want to adjust for include age, gender, education level, race, able carrier status. Next is about the statistical analysis. For descriptive analysis, means standard deviation were used for continuous variables and the number of percentages used for categorical variables. Then we modeled the longitudinal change in cognitive factor scores using mixed effect model with subject level random effects for intercept and time. In such mixed effect models, time is the years from baseline, and the time coefficient represent the annual rate of change in cognitive factor score after baseline. And the retest effect coefficient was interpretable as the jump in cognitive performance between the first and later visit, after accounting for the anticipated changes due to aging. Both unadjusted and adjusted models are fitted. Then we include interaction terms between retest effect and the features of interest in the model. P-value less than 0.05 is considered statistically significant. One consideration for this project is due to the COVID-19, cognitive tests were assessed by phone with a telephone-based adaptation of the cognitive battery, mainly at year 2 visit. This change potentially introduced mode effect, which could impact the estimation. So to minimize the potential bias due to the mode differences, the primary analysis was restricted to the in-person assessment conducted at baseline, year 1, and year 3. There are a total of 977 participants with 2,931 visits. And sensitivity analysis were conducted using a subset of 33 participants over 252 visits who completed all for assessment in person at baseline, year 1, year 2, and year 3. Another consideration is in a Chi May paper, a sensitivity analysis showed a significant difference in the effect of hearing intervention on cognitive change between Eric and de novo subgroup. So we are concerned whether there is any difference in the effect of hearing intervention on retest effect between Eric and de novo. To answer this question, we stratified the total participants into Eric and de novo subgroups, and the stratified analysis were performed by population source. Means Eric first de novo to examine whether the interaction between a trial intervention and retest effect differed across the subgroups. Here I show the result. Table 1 shows the baseline characteristics of the sample for primary analysis, which includes 977 participants. These Table 1 results are much similar with the Table 1 in the published original Achieve paper. The sample had a mean baseline age of 76 years, with about 53% being female, 87% identify as white, and 53% holding a bachelor's degree or higher. Additionally, 29% were APOE4 carriers. And 24% participants were recruited from the Eric study, and 75% participants were recruited from the de novo. participants were were evenly randomized to two intervention groups, 50% assigned to the hearing aid intervention and 50 assigned to the health education control group. At baseline, at baseline, about 56% had Pune average between 30 to 70, and about 43% had Pune average between 40 to 70. By design, baseline means factor scores for global cognition, language, executive function, and memory were all 0 and had standard deviation slightly smaller than 1. Here, table 2 showed the unadjusted results from random effect regression models among participants with baseline year 1, year 3 in-person assessment. And here, table 3 showed the results after adjusting for the demographic covariance. We could get the same inference from the unadjusted and adjusted regression models. The results show that there are significant recast effects in global cognition factor scores, executive function, and memory factor scores, but not for language. We could also see that there is a smaller recast effect among participants with poor baseline hearing level in global cognition, executive function, and memory, not in language. And de novo population have a stronger retest effect than average population in global cognition and executive function, which is what one could expect given that average participants have already taken this test before. There is no significant difference in retest effect by intervention, which could imply an effective randomization. For sensitivity analysis, we use data from participants with complete 4 in-person visits. There are 33 subjects included. Table 4 is the unadjusted result, and table 5 is the result after adjusting for the baseline covariance. The same inference could be obtained from adjusted and adjusted models. We could see that although there are only 63 participants including the analysis, it still shows strong retest effect in global cognition, executive function, and memory, but not for language. And also, people with poor baseline hearing level show a smaller retest effect in global cognition and executive function, which is consistent with the findings from primary analysis. However, different from the primary analysis is there in this sensitivity analysis, de novo population do not show significant difference in retest effect compared to average population, which could maybe due to the less sample size. Table 6 shows the adjusted result from random effect models after stratifying the total 977 participants into average and de novo subgroups. We could see that there are significant retest effects in global cognition, executive function, and memory, no matter in the average subgroup or de novo subgroup. What interests here is that among average subgroup, those in the hearing intervention group demonstrate a significantly greater retest effect in global cognition and memory compared to the control group. In contrast, no significant difference in retest effect by intervention were observed within the de novo population, which suggests a different hearing intervention effect on retest effect between average and de novo group. Finally, I want to conclude the findings. Our study demonstrates that retest effects are significant in global cognition, executive function, and memory factor scores, not in language. And the magnitude of retest effect should be comparable to 1 year of decline in age-related cognitive performance. There is strong retest effect in the de novo population compared to the average population, and there is smaller retest effect among participants with poor baseline hearing level. Overall, there is no significant difference in retest effect between the hearing intervention group and health control groups, but we could observe a greater retest effect in hearing aid group within the average subcohort, not in de novo subcohort. To conclude, by using the data from the CHIP study, we demonstrated the retest effects are substantial and can vary significantly across key subgroups such as baseline hearing levels and population sources. Our study provides insights into the magnitude and the variability of retest effects in longitudinal cognitive aging research and highlight the need for robust methodologies to account for this effect to ensure accurate estimation of cognitive trajectories and exposure effects. Future research should further explore the strategies to mitigate the retest effects through both study design and study design strategies and statistical approaches. That's all. Thanks for listening and your comment, discussions, suggestions.
Great job, thank you. Any questions?
So just one comment. And that is that the magnitude of the retest effect is comparable to the 1-year decline, as she pointed out. And that means really that you have to either be looking at 2-year data or you have to do some pretty substantial adjustments to the retest effect whenever you look at anything, because they're of the same magnitude. And I think that's a pretty important observation for anything that looks at cognitive decline using this set of testing tools.
The thing that because of the COVID-19 influence, there is only about 33 participants taking the in-person neurocognitive test at the year 2. So it's a limitation for this study to.
That's fine, but my point is the implication of this, not the logistical issues related to COVID, but the implication is that over time, your retest effect is the same as your 1-year decline in magnitude. And so it's important to account for that, because otherwise you'll reach erroneous interpretations. And so I think it's a very nice and important piece of work.
Yeah, so maybe for the Achieve main paper, when they evaluate the trajectory of the cognitive decline, maybe they could use some statistical method to account for the retest, like to use the use this binary variable to indicate whether this is the first assessment or the subsequent assessment to account for the retest effect. I think it's a good point.
Any other questions? So one small question, given that we know this from the study, how might we change testing procedures, testing policy? What would you recommend?
There is some papers that suggest that we could, at the baseline assessment, we could test the person twice to average the score to minimize the retest effect.
Great ideas. All right, if there are no other questions, I think we can adjourn for today. Thanks, everyone.